Welcome back to this deep dive. We are exploring a fascinating new paper today about rejuvenation. And this study isn't just about maybe delaying things a bit, it's really tackling the ultimate challenge, the reversal of age-related decline.
Hey, everyone. Yeah, this research is, well, potentially a genuine game changer. They focused on a therapy that works after the damage is done, so to speak. We're talking about successfully reversing frailty in animals that are already old, already slowing down.
Yeah,
Specifically, mice that are basically the biological equivalent of 75-year-old humans.
that distinction is huge, right?
Yeah.
We should pause on that for a second because, most previous longevity methods were really preventative. You think about rapamycin targeting the MTOR pathway. It was impactful, sure,
Mhm.
but it showed mixed efficiency. Results varied a lot between studies, and there were pretty serious safety concerns. Its immunosuppressant nature links to increased cancer development.
Yeah, so
This new approach kind of throws that paradigm out the window. They're actually treating a pre-existing condition, frailty, using this unique two-part therapy, targeting the whole systemic environment, like the body's operating system.
it's the ultimate stress test for any longevity molecule, really. And the results, well, they're pretty stunning. We'll definitely get into those numbers soon. But first, this breakthrough really comes down to the molecular architecture they used. And speaking of support, we must thank our sponsor for helping us dive into these complex topics.
Absolutely. We are really grateful to be supported by RevGenetics. They've been championing health and longevity since way back in 2007, providing high-quality longevity supplements. And remember, you can catch this deep dive on Spotify and also on YouTube, so listen or watch wherever you are.
So the scientists behind this, they realized that aging isn't just about one thing going wrong. It's more like a whole system falling out of sync. They focused on something they called simultaneous calibration.
Simultaneous calibration.
Yeah. So instead of just hitting it hard with one drug, they aim to use two components, things that influence the body's aging signals in opposite but complementary ways. The goal was to recalibrate the entire system back towards a more youthful balance.
Huh. It sounds almost simple when you put it like that, but I bet the biology is complex. Let's break down this double whammy then. Part one.
Part one is oxytocin, OT.
Ah, the love hormone. Most people know it for that, right? Social bonding and all that.
Exactly. But here's where it gets really interesting for aging research. OT levels, they naturally decline, quite dramatically actually, as we age. And beyond bonding, OT turns out to be absolutely critical. We're talking maintaining muscle strength, bone density, regulating healthy metabolism.
Wow. Okay, so that's the good signal that's fading away and they want to boost it.
Precisely. And part two is the essential countersignal, the ALK5 inhibitor, or A5I.
A5I. Okay, so this one's the blocker.
That's right. It inhibits a really crucial pathway called TGF-4 that's transforming growth factor beta. And this pathway, it becomes overwhelmingly active, you could say overpronounced as we get older.
Okay, TGFBO. Why is that the bad guy here? Can you make that a bit more relatable?
Yeah, absolutely.
Yeah.
Think of TGF-4 like this. It's the signal your body uses to tell tissue, okay, time to scar over, maybe shut things down. It's pro-fibrotic.
Pro-fibrotic, meaning it encourages scarring.
Exactly. It actively promotes inflammation and fibrosis, which is that dense, kind of debilitating scarring in vital organs. Now, in a young, healthy body, it has a role in repair. But in an older body, it just runs rampant. It starts interfering with the ability of tissue stem cells to regenerate. It's basically telling healthy tissue to retire prematurely.
That's a great analogy. Makes sense. So, okay, they're giving back the good signal, the OT, that's diminishing. And at the same time, they're blocking the bad signal, the TGF-4, via A5I, which is increasing.
You got it. This dual mechanism, it sort of re-engineers the whole systemic environment.
Yeah.
And it had already shown some promise in shorter trials. Even just a quick 7-day treatment showed it could rejuvenate muscle, liver, and even brain tissue.
Just 7 days. Wow.
Yeah, they're basically nudging the body's entire environment back towards a youthful state. Now, before we reveal the really stunning long-term results this had on those old, frail mice, we just want to take a quick moment to thank our sponsor one more time. We really do appreciate the support from RevGenetics. They've been dedicated to providing quality longevity supplements since 2007.
Yeah, and whether you're enjoying this deep dive into molecular biology on Spotify or maybe watching us break down the data on YouTube, remember that RevGenetics is there supporting health span goals. Definitely check them out.
Okay, let's get back to it. The old, frail mice, these are the 25-month-old ones. They get the cyclical treatment, OT plus A5I. 2 weeks on, 2 weeks off. And the goal, remember, wasn't prevention. It was reversing decline after frailty was already there. The results were, Well, dramatic, especially and powerfully in the males. Yeah, I mean, just looking at them, the visual improvements were noticeable. The treated male mice, they looked healthier, better posture, just generally less frail. But the hard data, you look at the survival curves, it just blew the roof off.
Mm.
Measured from when the treatment started when they were already deeply frail. The old male mice saw an absolutely astonishing 73.73% increase in additional life.
73%.
Yeah.
That's incredible. Can you put that into days or something?
Yeah, they lived on average 221.1 extra days. Compare that to 127.3 for the control mice. Statistically speaking, the control males, their chance of dying, the death hazard ratio, was nearly 3 times higher, 2.868 times higher than the treated group. It just dramatically boosted the animal's resilience against, well, aging.
And crucially, this wasn't just about tacking on sick days at the end, right? This was quality life.
It's exactly right. We're talking dramatically improved health span. They showed measurable improvements in endurance, like on the treadmill test, significantly better physical performance, grip strength, agility, things like that, and even cognitive improvements, better short-term memory in tests like the novel object recognition test.
So it fundamentally rewound their physical and mental decline.
Amazing.
And this is the complex twist you always seem to find in longevity science. Aging is profoundly individualized. The female mice did not show these same robust benefits, not in lifespan, not in overall health span. It really demonstrates these clear sex-related differences.
Right, so it didn't work the same way in females.
Which really emphasizes the therapeutic strategies, well, they have to account for these biological differences between sexes.
That's fascinating, though, because the treatment was doing something in the females, wasn't it? You mentioned the acute systemic rejuvenation happened in both sexes. And didn't they find something about fertility, too? Yeah, that's the intriguing part. When they treated middle-aged female mice, so 8, 12 months old, the therapy actually increased their fertility. They had more weaned pups. So the drug combination is biologically active in females. It has effects. But that big lifespan benefit just vanished in the older cohort.
Okay, so the acute results seem key here. You said 7 days showed youthful restoration in both.
Exactly. The systemic proteome, that's the collection of newly made proteins circulating in the blood. They use metabolic proteomics to track this. After just 7 days, the treated old mice, both male and female, looked chemically younger. And critically, the treatment significantly diminished protein noise.
Protein noise. Remind us what that signifies again.
It's seen as a really reliable biomarker of aging, basically indicating general cellular dysfunction and messiness. And the treatment reduced this noise in both sexes initially.
So the initial fix worked beautifully in males and females. What happened long-term with the females then? Why did they lose the benefit between 7 days and, say, 4 months? Did the dosage maybe just suit the males better?
That is the pivotal question this research throws up, isn't it? The long-term data, 4 months out, that's where you see the paths diverge. Only the old male mice kept showing that beneficial modulation of their systemic proteome. The females, over time, seemed to become unresponsive, which suggests, yeah, maybe a complex interaction with the female hormonal environment, or maybe the dosage wasn't optimal for them long-term, like you said.
Okay, so let's focus on the success story then, the males. What exactly was being recalibrated in them over the long haul that kept them going?
Well, in the male mice, key signaling pathways, the ones that really control major age-related problems, were profoundly normalized. Youthfully reset, you could say. We're talking fundamental networks governing chronic inflammation, how cells handle stress, tissue breakdown. JAK-STAT, MAPCALM, age-range networks for those interested in the specifics.
Okay, so major system-wide adjustments.
Exactly. This robust, continuous recalibration seems to be what allowed their system to stay stable and resist decline. It strongly suggests a systemic reduction in chronic inflammation and senescence that's cellular aging. And that's really the bedrock of improved health span.
And this systemic anti-inflammatory effect, it links back perfectly to the blocking part, the A5I, right? Inhibiting that overactive TGF-A seems key, especially for fibrosis.
Precisely. We know that out-of-control TGF signaling leads to those severe fibrotic pathologies, the scarring in organs like the lungs, kidneys, liver, real hallmarks of aging. So by using A5I to keep that scarring signal in check, they seem to be allowing the whole animal to restore vigor and function.
It's quite elegant, actually.
And what's also really important here is the potential clinical feasibility. This isn't some super complex, far-off tech. Oxytocin, it's already FDA approved, used all the time in clinics. And A5I drugs, like one called Vactosertib, they're already in clinical trials for things like cancer. And so far, no major adverse side effects have been reported. This makes it highly translatable.
Yeah, that's a huge plus. It's not like complex blood exchange procedures or anything. It's a potential drug combo.
Exactly, a combinatorial drug therapy.
So this really positions OT plus A5I as a very promising intervention. Specifically, it seems, for extending male life and health span. And it distinguishes it quite clearly from other approaches. We mentioned rapamycin earlier.
Which actually showed better results in female mice generally.
Right. And then metformin, the results there are still kind of all over the place. Very controversial. Depends on strain and sex. The power and specificity of OT plus A5I for reversing frailty in these male mice really makes it stand out.
Which leads us nicely to our final provocative thought for you, the listener, to consider. It builds right on that sex difference. Since the female mice did show those acute benefits, the systemic rejuvenation, the improved fertility when treated earlier, but they didn't get the big lifespan boost later, does this mean the timing, the when of an anti-aging intervention needs to be fundamentally different between the sexes?
Like maybe females need intervention earlier or differently.
Possibly. Or, given that the long-term protective effects just seem to vanish in females, does achieving true longevity extension in the female body require a completely different strategy? A different drug cocktail, maybe? Compared to males, is their hormonal landscape neutralizing the effect over time?
Lots to think about there. Definitely something worth mulling over as this research progresses. Thank you for joining us for this deep dive into this dual approach to reversing frailty. We'll catch you next time.